Everyone, thank you for coming. Thank you to Dean Cooper for the introduction, and also to Dr. Singh from the Alberta Epigenetics Network for having sponsored this evening tonight. So of course at the university I teach, and I actually teach the introductory biology course, which is a very large course. But I have to admit, I'm a little bit more nervous tonight than I am with 200 students. But we'll see. We'll see what type of crowd you are. We'll see how many will sleep during this lecture. Okay, and I hope not. I'll try to make it interesting. I'm very happy to see a number of people here. I actually have my family here. My wife Sophie and our two daughters. I'll be calling her Sophie, but she actually is a scientist at Lethbridge College, and so Dr. Kearney. And the two of us actually started this project, and I'll be telling you a little bit about how we started it. I also have my mother who is here tonight. So I was born in Lethbridge, and she's here with her friend John and my two older sisters, and so I was the youngest, and so that might explain a few things as well. But again, and friends as well, and with whom I grew up, and I'll have a bit of a story about that too. So first of all, thank you very much for coming, and I'll start. So this is a photo that I took of chemicals that are in our laboratory right now. And what we're doing is we're doing basic research, but a lot of people ask us this question because we're actually looking possibly for what might be a cancer drug. And what comes to this is there a chemical that we have in the lab which might actually one day become a useful drug that will actually help people who have cancer. And if you're a person who bets, in fact, the odds for this are very, 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 very low. There's a 99.99% chance that the compound to which that arrow is pointing, which is right here, that the answer is going to be no, this is not it. But our job as scientists is to try to think our way through, because the worst thing that we could do would be to have a good chemical in our hands and then actually and miss it. Because once you check and you say it isn't, well, that's worse than it's a mistake you really want to make. So our idea is that if the answer is yes, what might happen is that might be a chemical that will save, and these are real numbers, that can save 1 million lives, that might create 10,000 jobs, and for pharmaceutical companies, it might actually be worth an awful lot of money. But we're not a pharmaceutical company. We're at the university, and another important thing will be is that we would learn something that's very, very important about human biology. And once that knowledge is there, then it's good forever. Okay, in this talk, so I'm going to say a little bit about my research background. I'll talk about the scientific background for what we call the Prairie to Pharmacy project. And I'm going to tell you a little bit about what we've discovered so far. And while putting this together, I was trying to look what would be maybe a common theme. And in fact, the common theme is going to be Lethbridge. And I'll point towards that a number of times in this talk. Okay, Lethbridge. I took this photo from my office window. I have a great office. It's it's because the university building is set. And I'm sure if you're from Lethbridge, you would know where it is. As many as you probably are here tonight, in the coulees, and it has this fantastic view over the city. I took it with a cell phone. It's a little bit blurry through the window, but it just really sort of captures our city. And one thing I always like to tell people is that Lethbridge is a science city. And my feeling is that there are a lot of scientists here tonight who probably know that. So we have the University of Lethbridge, we have Lethbridge College, the Lethbridge Research Centre, the Canadian Food Inspection Agency. We have a number of businesses that are involved in science and agriculture-related topics. And the city also hosted the Canada-wide Science Fair a few years ago, where we had 400 children from all across Canada. And the reason why we were able to host it is because there is so much science in this town. And I just think it's always important to state that, because amongst the other qualities of the city. But does everyone know this? So who is this photo? It's not me in 20 years or anything. Do you? Okay, I may have heard the name. So this is, right, yeah, some people are nodding, good. So this is Bertram Brockhaus, who passed away. He won the Nobel Prize in Physics in 1994. And he was born in Lethbridge. And there's not too many cities in Canada, or anywhere in the world, of course, who can claim to be the birthplace of a Nobel laureate. His family was actually from the Milk River area, but he was born in Lethbridge, and we have this, so we do have a connection for it. And just to highlight that Lethbridge really is a science city. Okay. I know a number of you in the room, but I wanted to make the connection between me and Lethbridge. And Dean Cooper actually said part of it is, I was born in Lethbridge. So how many people are born in Lethbridge? I know some of you are, and some of my friends in the back there. Yeah. Okay, and if you're born here, I think one thing I find, it is a reason just for liking it. You tend to like the area where you were born. And I do have this strong, I just like Lethbridge. You know, what can I say? I was born out of all the places where I was. And like many who were born here, I went to the University of Lethbridge. And I actually remember when it was opened in so this would be the main building in 1972. My parents took us, their children, to there, and it was partly because my oldest sister won an award. She was in Girl Guides, and she won an award, and that award was being given during the grand opening. And I remember I was 10, and I remember walking in the main hallway on the 6th floor and being very, very impressed by it. And I also remember the university students who were there. They were sort of flopping around. You know, this was the 1970s, like they owned the place, right? And things have changed a bit. I think students have gotten very, very serious for it. But so I took a first degree in biological sciences, and then I did a master's at the University of Calgary. And then I have a PhD in biochemistry, and I was very lucky. The person with whom I did my PhD, he actually got a Nobel Prize. His name is Sir Tim Hunt. He was later knighted. And it was an excellent lab to be in. I enjoyed it very much, but many of the things that he would teach us, so teaching was a big part, we still use in the lab today, you know, and that's getting on in time. And then I got a number of jobs. They're actually postdocs in the jargon of the field. One of them then was in Switzerland. Then the other, and that's actually where I met my wife, Sophie, you know, a neutral country. She's from France, I'm from Canada. And then the Institut Curie in Paris, which was a very important research hospital and institute. And then I went to the pharmaceutical industry. And it was in 2007 when we came back to Lethbridge. So in some ways, for me, it's not so long ago. And all of this was important for the things that I'll be telling you about today. And I wanted to say this part as well, partly because I find that the students today are a lot more nervous about the future than I ever remember myself or my friends were. And I know there's some students here. We have some students from my lab, which I'll introduce in a bit. And the way that I got here back to Lethbridge was I always liked science, so that part was okay. But there wasn't really a clear plan. I wasn't very efficient about it, and I'm not sure. I don't think that was a problem, because I was always learning things along the way. So if it really was that one thing led to another. You know, in my education, a big event in my life was I did what's called an independent study at the University of Lethbridge. And what that is that the students can go into a laboratory, and that will count as a course, and they will be taught and trained, and they'll gain experience in research. And I still do that in my lab today. And out of the many courses that I took, when I was a student, many of them were very good, but this was the one that actually probably had the most impact upon me. And then after that, because of that, I was able to get a summer job in research in Florida. Can you imagine? I was 19 on the Gulf of Mexico doing science. It was great, because I always wanted to leave Lethbridge, by the way. Yeah. But I came back. And because of that, there was a professor there who was running a summer school in Boston, and then he offered me to go to Boston the next summer and take his course, just because I was interested and because he was there. And then because of that, I was invited to come back as a student instructor the following summer in the Boston area. It was in Boston area that I actually met the person who would be my PhD supervisor in England, because he just goes, well, you really should, you need to, if you're interested in the cell cycle, you should come to my lab. So I said, where are you working? He goes, well, Cambridge. Have you heard of it? Oh, yes. Okay. And then, and with that, then I was able to work in a very, very good lab in Switzerland. And then, as I said, that's where I met Sophie. And she had connections. She was actually already had a job offer at the Pasteur Institute in Paris, right? The Pasteur Institute. And she was able to introduce me to someone who was then moving to the Institute Curie. And so then I was able to work in that person's lab. And the Curie Institute is very well known in France. And then I had to apply, of course, for jobs all the time, but I was able to get a job, which was quite hard to get. It's always hard to get jobs, but I was able to get one there, and that's how that worked. But I think one thing that helped a lot, and I guess really have to thank my parents for this, I would always work a lot. And I can say that not to brag, but my mother, my father, so they had a construction company here in Lethbridge. And I always felt I had to work in that at a very, very young age. And I used to complain a lot to my mom especially, saying, oh, it's so hard. But they just told me to keep sticking with it. And I think that actually helped a lot. Some of these things were actually quite hard, but just the ability just to put your head down, or as I sometimes say, just sort of shut up and work and try to get things done, actually turned out to be pretty helpful. I'll say this too, a little bit for the students, is what you do matters. And that's why I think the U of L is a great place with the liberal education. I never knew that I would end up back in Lethbridge. But I was always taking courses at the U of L that actually helped a lot. And one of them, and I have two examples of this. One is bad and one is good. The bad one was I dropped out of French 30 when I was in high school because I did not see the point of learning the French language. And if you've noticed on here, I actually lived in French-speaking countries for 15 years, Dumb, but I didn't know. What was good is that I made the decision to do an independent study, and that sort of got me on all this. Okay. In terms of research, this is the project that I'm going to be telling you about today is somewhat recent. We actually started to work on it, Sophie and I, in 2013. The idea came before that, but in fact, everything that we had done before that, all of our training was then thrown into this project, and it's turned out to be pretty helpful. The basis for starting this project was from a number of things. Scientifically, we need more knowledge about how cells function. So cells are the basic unit of life. And a lot of things that are very important in living, just in health and in disease, are at that level. We need new cancer drugs. I had worked in the pharmaceutical industry, and then this was one that came to me as a bit of a surprise, was that the plants in southern Alberta have not really been tested for anti-cancer activity, and I was never quite sure about that. The other part was that the University of Lethbridge provided pilot funds to start this. And what they do is that they have competitions, and in some ways it's not a lot of money, but when you had no money for a project, it turns out to be a lot of money. And it was just the fact that they ask for new ideas, and then they evaluate them, and then we were able to get some of that, and that's what got us started. I'm not going to say a lot about cancer. I'm not a medical doctor. But and I think a lot of what I would say is actually known. It's a terrible disease, something that we really need to do an awful lot of work on. But just about the impact. So this is from the Canadian Cancer Society. In 2016, there will be 200,000 new cancer patients in Canada. Okay, Lethbridge has just reached the 100,000 mark. So every year, twice the city of Lethbridge in Canada, there will be new patients, right? This is still a pretty serious disease. I was just before this, I was interviewed at the University of Lethbridge, and one of the questions that came up, well, people have been working on this for so long, why isn't there more progress being made? Which is I think, a fair question, but it's also a very hard question. Improvements are being made in prevention, in detection, and in treatment. So things are getting better. I would say, especially in leukemia, really tremendous progress has been made. But just that it's a very, very tough one to crack. And we still need better, and we need safer medicines for treatment. And that's part of the thing that we're trying to do in the lab at a basic research level. Okay, there's a number of ways to approach it. And many of them are very valid ways. The one that we'll do at a basic science level is as a concept, is quite simple. Is that if you can find out what's wrong with a cancer cell, you will be able to fix it. Okay, it's the same thing when your car is wrong, when there's a problem with the car, you take it to your mechanic because your mechanic will know how the car works. And then he or she will be able to fix it because they'll say, this is the part that's broken, this is what we need to fix, right? It might cost you a fortune, but you know that you will be able to get that done. And we're not at that stage yet with cancer cells. We still don't quite know what is it that will make the cancer cell different from the human cell. But of course we know the outcome, although there are some good exceptions to that. In order to get that, we need to do more research, so we keep asking for more. And we also need to do more training because we're at a point where these questions are scientifically and technically quite complex. And so it does take years to train people to get to that point where they can start being useful to see further and see better than the current generation of scientists. The other part is then the challenge is once you know what is wrong, is to find a chemical that can fix it. And there, actually, the science is better. There's a number of examples of when the problem is found by a biologist, the chemist can make the compound. So we're quite optimistic on that, and we're trying to do both of these at our level in the lab. Okay. Conceptually, again, things are simple, but the grunt work is somewhat difficult. So one of the goals in the laboratory is to discover new chemicals with anti-cancer properties. In order to do that, you really need two things, because all the words count. You want something that is new. Research is always about being new. In order to find new chemicals, you need to have a source of where those chemicals come from. And then when you have all those chemicals in front of you, like I showed you in that first slide where I had all those tubes, you need something to tell you which is the good one, so that you're not guessing with these terrible, terrible odds, that you probably will be wrong if you were just to guess. And those are the approaches that are used we're trying to use in this study. Okay? Okay, I'm going to just check the time here. Okay? Fine. So why look for medicines in plants? Because plants have always contained important chemicals for food and for health, right? We eat your vegetables, right? So my mother's here, I can say that, right? We need plants to survive. They actually have all the things that are inside them that will keep us healthy. But more than that is that plants have a special capacity to make chemicals. So the chemist can make chemicals, but nature can make them too. And sometimes nature can make some that work very, very well and that do precisely the thing that we want. And then another reason, which I'll come to a little bit later, is that, and we know how to grow plants. And I guess I certainly don't have to say that here from southern Alberta, right? We are an agricultural region. Okay, more about plants. So plants, they make a class of chemicals, which this is a scientific term, although we use it a lot in the public, that's called natural products. And sometimes natural products make very, very good medicines because they're actually made to work with the living. Just to know, So this is a little bit against some of the advertising. Not all natural products are good for you, of course, right? And sometimes marketing will say that, but some of them are very, very good for you. In fact, about half of the modern anti-cancer chemotherapy, so more than just a little over 50%, are natural products or they're derived from natural products. Once again, it's been, nature has made some very, very good chemicals. And sometimes pharmaceutical industries have been trying to make better ones synthetically, but it hasn't quite worked. And now there's a realization of that. Again, I've worked in industry, and they're coming back to natural products. The example, so when I was giving you those numbers right at the very beginning, this is one which is Taxol, which is widely used in breast cancer. 1 million lives have been saved thanks to this. That's people who have been saved by it. Enormous amount of jobs. And for the company that made it, they actually made a lot of money. There's a lot of money that turns around that. So a whole variety of reasons. Then number 2 is another natural product which comes from plants. This is from plants and this one as well, camptothecin. I'll say a little bit about Taxol. So that was the first one. The best cancer drug. Natural product. It comes from a plant. So the Latin name, Taxus brevifolia, and we actually have a sample of it on the table just at the entrance. And it actually grows. It grows in Canada and in higher altitudes in the U.S. It's called the Pacific yew. We were actually able to get a sample of it for testing in our laboratory from Mr. Charles Filovich, who he's a friend, and he also went to the U of L, and he lives in West Vancouver. And it was discovered in the United States in 1971. It took a number of years to actually make it into a pharmaceutical drug, and it gave rise to Taxol, or the Taxol, which is the brand name. And it also gave rise to a second idea, where in France, a related plant, Dr. Pierre Potier, found a somewhat similar chemical that did the same thing, which saved another 1 million lives from this work. A touch of science, and what I just want you to know is that we were able to get that here in Lethbridge, and it was very important for us to start 4 years ago. Because what we wanted to do, we wanted to say, if we can get the best plant that is out there, we should be able to detect it in all of the tests that we're going to set up for using for other plants. And I guess we were able to compare it with the original stuff which we were able to purchase, and then the plant which we were able to extract in the lab, and it gave all sorts of clues that we were able to detect it. And that was a proof of principle validation step. And with that, I was able to go to the competition at the University of Lethbridge and ask for pilot funds to get us started. Because I had to demonstrate that we might be able to deliver on our promise to find something new. Okay, so plants. Now, why investigate plants in Canada? Because the odd thing is that Canada is not commonly recognized as a country in which to look for new medicines, right? The Amazon, Costa Rica, French Polynesia, anywhere where you would see plants probably is where normally most of, there are many scientists who are looking for plants, there are many companies who are looking for plants. And this is in the field, this is a very famous book because the person who wrote it discovered not one, but 2 very important cancer drugs, which is, in fact, he's the only one who's ever done that. And he wrote a book about it, and in fact, he lists 80 places on the planet to look for cancer drugs, and Canada is not on that list. So the idea of looking for new medicines from plants is absolutely not new. But the idea of looking in Canada, in fact, turned out to be somewhat new, much to our surprise when Sophie and I started this. Of course, here's why. To most other people on the planet, they think of Canada as a cold place that's covered in ice and snow. And having lived outside of Canada, they're right. In fact, the weather is better almost anywhere else, even though we love it, right? Here's a bright sunny day, it's about minus 22, and nothing is going to grow there. So that's probably one of the reasons, in fact. But while working on this topic and having connections with France, I learned something, which is that the history of Canada was actually guided by a medicinal plant. So you have to here's part of your liberal education. We have to go back a little bit. And so Jacques Cartier, who was French, and sent by Francis I, who was the king of France at that time, to discover the New World, because Spain had already discovered the southern part. See, so he sent Jacques Cartier, who went on 3 trips to the land of which is now Canada. It was on the second trip, they were going up the St. Lawrence Seaway in the month of October. And of course, the internet hadn't been around yet, so they did not know that October was going to get very, very cold. And there were 3 ships, and those ships were caught in the ice, what is near of what is now Montreal. They did not know that ice was going to stay there until the end of April, and they were stuck there for the end of April. What happened, though, is that, so this is the museum, this is his house. When I dragged my children there to visit it once, and so we actually went to see it. And so the original documents, sort of like captain's log, star date, whatever, are there. So he actually wrote, and what he wrote was that in the month of December, they started to become very sick. Okay, it was quite serious. And already 25 members of 101 seamen had died, and 72 or 73 were very sick, and only 3 of them, and Jacques Cartier himself, were actually okay. They were dying from scurvy, and they had no idea. 1, because they didn't bring any plants with them to eat vegetables, and 2, everything was frozen. And in fact, it was the First Nations there who realized what a terrible state these newcomers were in. They actually gave them, they recognized the disease, and they actually gave them leaves from the eastern white cedar, and it cured them, of course. Because when you have the right medicine for the right problem, it solves it. And then the rest is history. They survived. Jacques Cartier went back to France. They organized a 3rd trip, and then eventually that led to, for better or for worse, as we say, for the colonization of Canada by France and then later by England. But they almost lost it at that time. And that brings us to this point. So when I say to you that this area has not been explored for medicines, of course I'm talking about the Western scientific view of it. Of course, the First Nations from this region and from other regions across Canada have actually survived for medicines and for health for the plants that have been in this region for as long as they've been here. So the laboratory is actually in contact with two First Nations communities. One is the Piikani Nation, and one of the starts of this project was actually a discussion which began with my youngest daughter and Mr. Conrad Littleleaf at the head smashed-in buffalo jump. And then we also work with the Kainai Nation in Canada, which is the largest, in fact, and in fact their culture knows a lot about all of this, and we're finding ways to exchange information about it. I like this photo a lot because it actually captures everything. So my lab has seen it probably 100 times, but... Okay, so this is a photo of southern Alberta. And in one way, it conveys the information of why very few people have scientifically looked for plants in southern Alberta. Because the first thing is, there aren't any. And I was born here, right? And so I knew that. But in fact, there are. And it took me a very, very long time to learn that. And so this was all taken on the same day. This is the Twin River Provincial Grazing Reserve, which is 150 square kilometers, somewhat pristine land, and it's it'd be about 30 kilometers west of Milk River. And on the date that I took this photo, these plants were already in full bloom. And I have wandered quite a bit in the coulees before that with my friends, and I'd never really noticed flowers before, and some of my friends are here tonight. And it's partly because we used to only do this at night behind the college, so we didn't really see a lot anyways. But even during the daytime, when we'd go back and clean the site up, I still didn't really notice a lot. But now I see them, and I've dragged my friends out to see the flowers with me. They're still surprised. So it's great. So there are plants that grow there. But even that is not sufficient as a reason to do this. Probably the biggest reason is that the only reason why there are flowering plants that grow on the prairies is because they are able to survive a competition with the grazing mammals. So this photo was taken by my brother-in-law. It's a great photo. It's sort of this dreamlike photo of the buffalo. These buffalo are very close to the Head Smashed In museum site. And for as long as there have been plants growing here, there have been mammals growing here. And you can imagine, as I've talked to you about the winters, right? A very cold winter, and now the first green plant is going to push up through the snow, and there's 100-kilogram buffalo that's been waiting for that moment for about 6 months. So if the buffalo eats the plant, then that's it, right? That plant will not flower and seed, and it's over. So the plants, through evolution, are pushed to make chemicals, if you wish, that will actually stop a grazing mammal such as this. And then the mammal adapts so that it can still eat the plant, and then the plant adapts so it will make a more stronger chemical. And it's been going on like that since time. And that's what makes this area, even though really there's very few plants here compared to the Amazon, that's what makes the plants here special compared to other areas. And it actually took us a long time. I think a lot of people knew that, but it took Sophie and me a long time really to figure it out and then sort of launch this project. So I keep calling her Sophie, but it's really Dr. Kearney's, right? So what happened is that we got to work, it was really 2011, and we spent really the first year of trying to figure out which would be the best plants to do it. And from that, so there's about, the plants in this area have been very, very well characterized. So they know which plants are here. And also we have the good fortune that there are also many, many experts on these plants. And I have one of the books here. June Flanagan is here, and she actually has a book and knows this area very, very well. So those data are there. And then it required sort of bench scientists such as ourselves to pour through all of that and try to figure out which might be the interesting ones rather than just trying to get them all. Okay, I'll check the time again. Okay, a bit of the sciencey stuff. So now there are plants. We think there are reasons to test these plants, and now we're going to do a scientific approach to find out what's inside them. And we try to do this, as I say, sort of the best we can. And what we do is we collect on undisturbed public and private lands with permission. Okay, we do what's called a sustainable harvest, and there are many guidelines to do that, so we're not ripping out the last one. It's not like Pokemon or anything like that, right? We always have to save enough so that we don't disturb the ecology. We collect voucher specimens, which are then provided to the University of Lethbridge Herbarium, which is an accredited, well-recognized herbarium that already has 20,000 specimens in it, and that information is available to the public. We isolate the DNA from every plant that we work on, and then we create an annotated extract library, which means that there is a lot of data that is going to be associated with that, and then we try to do the best we can for a controlled experimental design so that if there is something in there, we'll find it. Okay, so this is a photo of one of the, this is not very far from Lethbridge at all, it's just by the airport area. So this is, again, some of the people who've helped us. We have Mr. and Mrs. Dale and Ingrid Morden. They let us go into their land, Mr. Robert Luco and Mr. Michael Monahan. Also, we have permission from the city of Lethbridge to go on certain areas within the city limits, but there's a lot of the coulees here are actually quite nice, and there's areas where we can collect from. And then from the Alberta government, the Twin River Grazing Reserve near Milk River, and then the Castle Special Management Area. Mr. Jeff Wright, who's in Blairmore, helped us with getting the papers for that. And then the work begins. And I've had a large number of people help us, so family, but members from the laboratory as well. Some of them are here tonight. Here we have Jan. What else? Here we also have Alessandra, members from the university, Professor John Bain, Joanne Golden, a former student, and then friends have come out as well. It's a fair amount of work, right? These are not cultivated fields. We have to go out and find them, collect them, and sometimes we only take 1 out of every 10 plant if it's particularly difficult to find, to ensure that there are others that are there. And then we prepare an extract library. It depends how you look at it. Sometimes maybe you need a lot. So this is about 10 kilograms of wet material. Collect that, clean it, sort it, and the idea is to eventually get that into an extract library, which is annotated, traceable, validated, and to date we have 42 unique plant species and from 17 plant families. And what I mean by this is they have not been studied elsewhere in the world yet. And that's some of the stuff that's actually growing, if you wish, in our backyard. And then you need a team. And so these are a number of students in the laboratory. I'll be talking about their work. And again, thanks to the University of Lethbridge, we're able to attract very, very, very good students. And you see, not all of them are from Lethbridge. Okay, we're also able to attract very good ones from Lethbridge, but from far away and far away too. Dr. John Bain, he's not really, I mean, he's at the University of Lethbridge, but he's been very, very helpful. He's a botanist who's helped to get us started. And we have a wider network, because we cannot do all the experiments ourselves, and partly because some of it is chemistry. A very, very important collaborator for us is Dr. Raymond Anderson, who's a natural product chemist at the University of British Columbia. We're quite fortunate. He's Canada's best natural product chemist. And we were able to interest him in this project. Sophie and I, we actually went to meet with him, very busy person, and just explained our story, and he agreed to work with us. We also have some members from some of the Alberta government scientists, Dr. Bandera, who is here tonight, who's a crop specialist, also others from here. And you'll notice what I'm doing on these maps, I always have to put the dot of where Lethbridge is. Okay, and I'm going to talk about that at the end as well. And then we're closely connected with laboratories in France, and that's very good because France has this tremendous expertise in natural product chemistries. They're one of there's 3 or 4 countries that have that expertise, and so we're quite lucky through our past contacts to be with that. So Sophie and I used to work together at the University of Lethbridge, and then she found a better job. She's at Lethbridge College. And so what we did is then we divided the project in half because both of us started it. And I won't say much about it because this will be hers. This is her story. I'm talking to you tonight about cancer, but there's also a very, very big problem with antibiotics and infectious disease. And she's just set up a laboratory, I guess a little over a year ago, where they're now using the same approach to find new antibiotics. Okay. So we're able to get chemicals out of plants here. The thing is, which one is the best chemical? And for that, on paper it's very simple. You just need now a biological assay that will sort of make the chemical speak. And that's our second approach. This is where we really use our past expertise, both from basic research and from the pharmaceutical industry. And that is, we use a special type of assay, which is called the phenotypic assay. And we use this because a number of reasons. One is the pharmaceutical industry doesn't really use this too much, and so we're not going to compete with them because that would be dumb, right? They have robots, they're very large, they're very, very good. And we try to do the things that are actually too hard for the robots to do. The other is we don't have an a priori, if maybe we don't know, in fact we do not know everything about what causes cancer, but if there is a chemical within that plant, we'll just let that plant tell us that maybe it can work. And in fact, historically, and it probably comes from because people keep asking, well, why aren't we being more successful? The whole cancer research field. And it turns out phenotypic assays are the best way to do it. Okay, which ones? So again, a lot of research out there, and we were able to, so we have a lot of guidance from other laboratories, is that there are a number of criteria for cancer cells that are called the hallmarks of cancer, and we pick from that, and here's the reference for it. Okay, so a little bit of data. So the project is actually working out quite well. Our first major plant of interest was the buffalo bean. It was interesting for us because it meets many of the criteria. It's actually found in very, very, very few other places in the world. It's just southern Alberta and northern Montana, and that's about it. Okay, not too many plants have that. What I'll show you here is that it's just we were able to do the experiments which helped us identify it as a plant of interest. And very early on, we were able to get in contact with a pharmaceutical company in France, Pierre Fabre, and we chose them because they are the best at finding chemicals that come from plants. That's called phytochemicals. They actually use it, they don't use this, but in Canada, they don't sell medicines yet, but they do a lot of cosmetics and shampoos, and some of those are in Canada. We were able to find out that data. We had our first publication, and we also had our first patent on this particular plant. And now in the laboratory, one of the graduate students, Jan Tuscher, is actually going the steps further on this particular project. I'm going to say a little bit more about this one, because these are our newest data, and I thought it'd be important to tell you about that, to say something new. And that's the Gaillardia aristata, or the brown-eyed Susan, and many of you might have recognized this. It's very striking. It's a very pretty plant. It grows, so it's not just southern Alberta, it actually grows in dry regions throughout North America. It is native to the zone in which we live in, typically not eaten by animals. Okay, so that was a very big clue. It had already been reported to be toxic in cell assays, but they didn't go much further than that. And it was also known in traditional knowledge, mainly from British Columbia, as being a medicinal plant. And this work is done by Alessandra Bosco, who's a graduate student who's just finishing. She's here tonight. And I'd like to highlight, it was actually privately funded by Mr. Michael Monahan. And that was very, very important to us, because it's early stages, and it's very hard to get funding for a brand new project. There's a lot of competition out there. We start all of these sub-projects with 3 questions that we must answer. And that is, does the plant extract have an anti-cancer effect? Yes, we'll work on it. And then we must know precisely what is that effect. Because we're going after the chemicals. We're not, in a lot of cancer medicines, they don't give extracts to people anymore. They want to know precisely what is the molecule, what does it do, and what is that molecule? And with that, we're collaborating with Dr. Raymond Anderson again from British Columbia. Okay, a bit of science. I'm going to show you these images because I think you'll be able to understand a little bit what we see. So we grow cancer cells in the laboratory. This is from a colon cancer, and we're able to image them with a microscope. They grow here. And then, and you can actually see them here. And I don't know what these blobs might look like to you, but there's, here's one. There's about maybe 10 here. And we call it PP006. When we add the plant extract to these cells, you can clearly see that there's a big change in the shape of those cells. And that was our first clue. We didn't quite know what that was in the beginning, but it told us to keep going forward. And then here's more. We then analyzed various parts of the plants and the various conditions. And now here, these small dots that are grouped together, there's about 100 of them. And there's about 100 here as well. But what you can see from this image is that this image is different from that one. And this image is different from that one. And we're able to then measure that and quantify it. And that tells us a whole bunch of things. First, it tells us that there's a particular part of the way that the cancer cells are responding to this. And this particular shape change is quite important. But it's also different from known. This is not a cancer drug, but it represents one, but it's also different from known ones. And so now when we did this, we thought we're seeing something brand new. And then it goes on. What I want you to do here is to compare the shape of this object here with this shape here, and clearly they're different. It actually measures a different part of the cancer cell. I know there's a lot of scientists here, so this is flow cytometry, and we're measuring the amount of DNA throughout the cell cycle. And I'll say that to those who are familiar with that, it actually will stop this likely at 2 different parts in the cell cycle. And that was an important clue for us as well. But again, even without being familiar with the technology behind it, every step of the way we get a very, very clear difference of what's going on. And then this is the last one. What you'll see here are different shapes and different colors. And what we were able to do is then we were able to meet with Dr. Raymond Anderson from UBC, a chemist, and he kept separating that crude extract with thousands of chemicals in it until we came with one. And then Alessandro did these experiments, and what we were looking for was there is a particular test in which we're actually a very high magnification. This is 600 times. And one part of the cell we can color green, and it will have this shape. But what we really want is this shape here. And we can get to the shape when we add the chemical, which comes from the Gaillardia plant. And it's a chemical that has not been seen before. So it's from a family that's known, but it's a novel member of that family, and it has a brand new effect that has not been reported before. And then Dr. Anderson's team, that's called hemisynthesis, they then took the natural product and they've made it even a little bit better. What I would hope to do, so none of this has been peer-reviewed yet. This is all brand new. What will happen is now these data will send them to people whose sole job is going to be to criticize it. Right? This is the hard part about science. You work hard and give it to someone, and then they cut it to pieces. But it's very important that they do that so that the work can be improved. And that's the next thing that we're going to do. We have quite a bit of confidence in the work that we've done. And now we have to send it to experts, and the experts are going to say whether they agree with it or not. But for example, Alessandro's worked on this for 2 years, and we're pretty sure that it's going to turn out to be right, and I'll let you know. Okay, I'm going to come to the end. So where are we going with this? Well, we've created a unique and a valuable plant extract. Okay, 42 plants, of which there are brand new things to find out. We're training the next generation of scientists. So we have some very, very specialized techniques, and we received an excellent grant from the Government of Canada, in which we have some excellent equipment now in which to train the students. And we're now going to a step, as I mentioned with Dr. Bandera, we're trying to see if we can actually grow these plants. And when they are grown, do the grown plants still have the same chemicals in them? Okay, with the idea, and I don't know if you've traveled in Holland and you've seen the tulips, or maybe in France and you've seen the lavender, that maybe one day in this area, we can actually grow the plants that are making the chemicals and get lots of them. And so I'm going to come to the end. So that's something that I'd like to actually say to this audience. Because after a lot of discussions, we believe that that Lethbridge in southern Alberta might have all the expertise that is necessary to discover, cultivate, and develop some of the chemicals, if you wish, that grow in our lab, and maybe even in this room. Right? There are people who are experts in agriculture, experts in cultivation, chemistry, and even in business. And it would be great to see if we can go to that next step. And, where I was putting Lethbridge on the map all the time, and maybe someday we won't have to do that. Because what I didn't say is when I was, while I was studying, I was going to all these different places, where are you from? You have an accent, yeah. Okay, I'm from Lethbridge. Of course, where's that? Right? And it'd be great to change that. It's a great city. Okay? There are many people to thank. What I told you is, of course, the work of many people. So members of my laboratory, also the University of Lethbridge. I've had a lot of support to get this going. Right at the very beginning, where there was the biggest risk that it might not work. Also from volunteers. Chloe is here. I didn't have her photo. But Chloe is a student who actually helped us collect plants as well, and Randy Piskuski and Paul Landry and members of my family. Collaborators, I've mentioned some of them. The conversations with the Piikani Nation and the Kainai Nation. Very early stages, but also very, very important. We recognize that we're harvesting plants that is also part of traditional knowledge and also have cultural value. And thanks for funding, which is absolutely essential to get this work done. Okay, and I'll stop there.